Okay, Assalamualaikum and a very good day. So today we are going to learn about the cellular glycogen. Okay, and we will learn this one first before we learn about gluconeogenesis. Gluconeogenesis happens after or in opposite of glycolysis. But because glycogen happens in between them, so we will learn about them first, just a little bit. Okay, and this lecture should be very straightforward. So this is the molecule of glycogen. As you can see, it is an extensive network of glucose bind together through two major bonds. The first one, the linear bond, we call them as alpha 1,4 glycosidic bond. Okay, they all have form at a linear binding of the glucose, and the second one is alpha 1,6 glycosidic bond, which only happens at the branch of the glycogen. This molecule is in fact it is similar to the starch that you probably see in the plant, but because it exists in the human, although the molecule is similar, their way of breaking down or forming them can be very different. So before we start the glycogen lysis and also glycogen synthesis, we will see why glycogen can be used as energy storage molecule. The first one, because of course fat is a better energy storage compared to glycogen. Okay, the first one, because fat they can contain unlimited energy, but glycogen they only have limited energy. Means that after the body finish forming the glycogen in the body, it will stop and start converting all the glucose to fat. This is because glycogen is very heavy. It can attract two waters from one molecule of glycogen. So if the body synthesizes too much glycogen, the body will be too heavy. Okay? But for the fat, because fat is hydrophobic, so it is very light. Okay? So the fat can synthesize fat unlimited until actually if you eat a lot, there will be no limit to the storage of the fat. Yes, that's why you can see people get very big if they eat too much. Yeah. But glycogen possess several advantages compared to fat. Yeah. The first one is fat cannot be rapidly mobilized in skeletal muscle. Okay. Fat usually they will be synthesized in the adipose tissue and also in the liver before can be used by the muscle cell. But glycogen they can be formed in the muscle cell as well. Okay, on top of the liver lah. Liver is the major or the primary place where the glycogen will be formed, but the glycogen in the liver and the glycogen in the muscle will be very different. Glycogen in the muscle is exclusively for the use of muscle only, but glycogen in the liver is usually used to stabilize the blood glucose level in the body. Means that the liver will sacrifice their storage in order to make sure all the other cells, the peripheral cells, can survive, okay, by providing the glucose. Because we know that glycogen can be broken down into glucose, right? Because it is a extensive network of glucose. Secondly, fat cannot be oxidized to produce energy in the absence of oxygen. Okay, means that the fat, in order for the body to use the fat, it must undergo aerobic respiration. Okay, but if the body try to use the glycogen, they can only undergo the anaerobic respiration, means during the exercise and so on. Third, the energy input is required to initiate fat oxidation. Okay? For glycogen, you don't have to supply the ATP in order to break the molecules, but for the fat, you need to supply ATP first before you can break them down into smaller molecule and can be used as energy source. And the final one is no conversion of fat to glucose available in the body in order to maintain blood glucose level. Important for tissues such as the brain. Okay, if you look further or in detail for this sentence, it means that the brain they always prefer to use the glucose first until when you reach one point where there is no more glucose available at all in the blood, then the brain will start to slowly change the preference to fat like fuel. Okay, in this case, ketone bodies from the fat. I think you heard about ketone bodies before when you learn about the biology in your matriculation. So what happens is, if you use fat as a source of energy, it cannot produce the glucose. It only produces the ketone bodies as a source of energy. Okay, so body always prefer to use glucose first. So, and especially brain. So if you don't have the glycogen anymore, then body need to switch to fat, which is not preferable. And it's usually the release of energy is much slower. So glycogen is used when intracellular level of glucose and ATP are low. So the body start to release the glucagon, which is opposite of insulin, to start breaking down the glycogen. Glycogenolysis is a degradation of glycogen. Muscle glycogen is fuel for muscle construction. Okay, means that only muscle can use them for themselves. But liver glycogen is mostly converted to glucose to bloodstream transport to other tissues. Means that liver will sacrifice their storage in order to make sure the other cells can feed on the glucose. Okay, although this is similar structure to the starch, but the hydrolysis is not as simple as starch in the plant. Okay, so basically the glycogenolysis involves three major enzymes. The first one is glycogen phosphorylase, and this we already take a look in the introductory lecture, whereby one phosphate group will be added to the glycogen in order to break them into one glucose. Second is glycogen debranching enzyme. Okay, as the name suggests, it is used to cut the branch point of the glycogen. Okay, so glycogen phosphorylase is for the linear part of the glycogen, and this one is at the branch point. And the third one is glycogen phosphoglucomatase. Okay, because when glycogen phosphorylase cuts, okay, I just draw a little bit so you will understand. So this is glycogen. Okay, I draw with touch. Okay, so this is glycogen molecule. Okay, and then it will reach at branch point. Right. So glycogen phosphorylase will cut at the linear point of the glycogen, and the debranching enzyme will cut here. But then it will produce glucose 1-phosphate. Okay, this glucose 1-phosphate cannot be used as fuel for the glycolysis first, because remember in glycolysis we got glucose 6-phosphate. Okay, so what happens is the body needs this phosphoglucomatase to convert glucose 1-phosphate to glucose 6-phosphate first before they can be used to produce the pyruvate. Okay, this one you can take a look more in the lecture of the glycolysis. Okay, so this is the mechanism. You can look at it slowly, but in general it will cut this glucose by adding 1-phosphate, okay, and produce glycogen N-1 plus 1 glucose 1-phosphate. Okay, and it will stop until 4 glucose molecule left before alpha 1,6 glycosidic bond. What does it mean by this sentence? Okay, yeah, you can take a look slowly, and you can also find YouTube video about this one. So what happens is basically it will cut all this glucose until it reach the branch point of 1, 2, 3, 4 glucose molecule, and it will stop. Okay, then what happens is the second enzyme will come in. Okay, the second enzyme will come in, and this debranching enzyme, the second one, will come and cut this one and transfer this one to this end of the linear molecule, the longer one. Okay, now you can see it is longer and this tree is gone. Okay, and now this glycogen phosphorylase, the first enzyme, can come cut again until it reach 4 molecules from the branch. Okay, until it reach here. Okay, then the last and then the second, after the glycogen debranching enzyme transfer these 3 glucose molecule to the linear branch, the second activity of the enzyme will come and cut this alpha 1,6 glycosidic bond. Okay, remember this one is 1,4, and then it will come and cut this alpha 1,6 glycosidic bond. That's why we call this one as debranching. Okay, because it can debranch the branch of the glycogen. Okay, and then when it cuts this one, it will add one phosphate here. So glycogen debranching enzyme, it will cut the alpha 1,6 glycosidic bond by alpha 1,6 glycosidase activity into a single glucose unit. Okay, it is quite special because one, it can cut a different kind of bond. Secondly, when it cuts this one and it add the phosphate group, this glucose will automatically become glucose 6 phosphate. 1, 2, 3, 4, 5, 6. So glucose 6 phosphate directly. So for this one particular glucose, you don't have to undergo the process of mutase or isomerase anymore. Okay, only the one that is being cut from the linear ones need to undergo another process of phosphoglucomatase to convert to glucose 6 phosphate. Okay, so the last one is the like I told you before, glucose phosphoglucomatase. Okay, the product of glycogen phosphorylase is glucose 1-phosphate. So it needs to be converted into glucose 6-phosphate for glycolysis. Note that this pathway uses less ATP than the entry of glucose into glycolysis. Okay, what it means by the sentence. Okay, because when we learn about glycolysis, we know the first one is glucose, and then it will become glucose 6-phosphate. Okay, and then it will be converted into fructose 6-phosphate, and finally there are several reactions until you produce the pyruvate, right? Pyruvate. Okay, so we know that for the first reaction, ATP needs to be used as investment phase. Okay, produce ATP. Sorry, it's not very good. So ATP converted to ADP, but when you convert the glucose, when you get the glucose from the glycogen, it can straightly produce glucose 6-phosphate. Means that it will bypass this first reaction. So instead of 2 ATP as investment, you just need to use 1 ATP as investment. So the total energy that we got from the breaking down of glycogen is 1 ATP extra compared to the normal breaking down of glucose. Okay, so if you remember, we know that there are 2 ATP being produced, net ATP being produced in glycolysis, right? But if you get the glucose from the glycogen, you will get 3 ATP instead of 2. Okay, and you see here in liver, glucose 6-phosphatase is used to convert into glucose in the blood. Okay, the one that we saw before is in the muscle, but okay, if the glycogen that we are breaking down is in the liver, like I told you before, liver is using the glycogen to provide the glucose in the blood. It will produce glucose 6-phosphate, but instead of being broken down into energy, it will go up, okay, and produce back the glucose. Okay, and glucose will enter into the blood. Okay, the process of converting to glucose needs glucose 6-phosphatase to cut this 1-phosphate, okay, and becomes glucose. Right, so this is the glycogenolysis. This is actually just the reverse reaction of the glycogen lysis. I will stop here because the limit, I already reached the limit of 15 minutes. I will continue in the next lecture. Thank you.